This short presentation is for those who are not necessarily scientists but want to understand the ramifications and consequences of a new study that calls into question present umbilical cord blood testing. For some years now, HemoGenix has been advocating improved testing for umbilical cord blood. This new peer-reviewed study published in the Journal of Translational Medicine emphasizes the need to phase out minimum testing and begin implementing best practices that provide quantitative measurements and trustworthy results that can be used by the transplantation physician to better assess whether an umbilical cord blood unit can be transplanted into a patient. First, let me mention what this study was all about. When a cord blood unit is processed prior to freezing, it is usually red blood cell and/or plasma reduced. This leaves cell impurities which do not contribute to the engraftment of stem cells even when the unit is thawed and used for transplantation. Since the functionality of the stem cells are not measured for a cord blood unit, the question arose, can the quality and potency of the stem cells be measured even if cell impurities are present? The scientific conclusions of the study can be summarized simply as follows. The answer to this question was a resounding no. Current processing procedures do not allow stem cell quality and potency to be measured. The presence of cell impurities masks and severely underestimates the functional ability of the cord blood unit. Segment samples produced at the time of processing are not only different from each other, but do not adequately represent the cells in the whole unit. The methods used to determine viability, that is, whether the cells are alive or dead, can produce a false positive result that does not correlate with the true viability and functionality of the stem cells. That is, viability may indicate a high proportion of cells that are alive, but the stem cells may be dead or cannot sustain proliferation and growth. The implications, repercussions, and consequences of these conclusions go well beyond this study. We can start in 2005 with the law to provide for the collection and maintenance of human cord blood stem cells for the treatment of patients and research, and to amend the Public Health Service Act to authorize the C.W. Bill Young cell transplantation program. This law was enacted in the United States and is also known as the Stem Cell Therapeutic and Research Act of 2005. It was reauthorized again in 2010. The Stem Cell Therapeutic and Research Act of 2005 provided funding for, among other things, the establishment of a cord blood inventory by the National Marrow Donor Program, NMDP, an advisory council for blood and stem cell transplantation which reports through the Health Resources and Services Administration, HRSA, to the Secretary of Health and Human Services. The Act also funded public cord blood banks to collect and maintain high-quality umbilical cord blood units. Let's take a look at the meaning of the term high quality, because it is critical to this whole discussion. The term high quality refers to the title of the statute, in which the stem cells in the umbilical cord blood unit have to be of high quality. Stem cells are of high quality and potency when they exhibit both the ability and capacity to proliferate respectively. So have high-quality stem cells been characterized in umbilical cord blood units? Worldwide, more than 600,000 cord blood units have been collected by public cord blood banks. More than 25,000 cord blood stem cell transplants have been performed from these units. In the United States, the government pays about $1,100 for each cord blood unit collected and permanently stored for transplantation purposes. And the average cost to the patient and/or healthcare provider for each unit of cord blood for transplantation is now estimated between $40,000 and $60,000. Since the first cord blood transplant in 1988, more than 25 years ago, it is safe to say that not a single cord blood unit has been characterized to ensure that the stem cells are of high quality, let alone high potency. As a consequence, umbilical cord blood testing in the United States has been non-compliant with the Stem Cell Therapeutic and Research Act of 2005. So what's the cause of this non-compliance? In its need to establish standards for unrelated allogeneic peripheral and umbilical cord blood hematopoietic stem progenitor cell products, the United States Food and Drug Administration, or FDA, designated umbilical cord blood as a drug in 2009 and provided non-binding recommendations for cord blood purity and potency. These are total nucleated cell count, or otherwise called TNC count, viability, and viable CD34 content. The term TNC refers to the state of the cells in the cord blood unit after red blood cell and or plasma reduction. This so-called TNC fraction not only includes the stem cells, but also different proportions of cell impurities, such as red blood cells that have not been removed, granulocytes, platelets, and other cell types that play absolutely no part in the transplantation process. Viability determines whether the cells are alive or dead. CD34 is a protein that's expressed on the surface of some cells and is considered a marker for stem cells, but the proportion of stem cells it detects is very small. Other regulatory agencies worldwide and standards organizations, including AABB and NetCord-FACT, which are responsible for the accreditation of cord blood banks, followed suit by requiring the banks to use these parameters, but also included a functional test primarily to determine the growth of so-called granulocyte macrophage progenitor cells, these are primitive hematopoietic cells, as a means to predict neutrophil engraftment after transplantation. This test is called the CFU assay and is falsely assumed to be a potency assay. According to NetCord-FACT standards, these standards are designed to provide minimum guidelines for cord blood banks. These standards are not intended to establish best practices. As a result, cord blood banks have no incentive or reason to improve umbilical cord blood quality and potency testing, and have therefore remained non-compliant with the Stem Cell Therapeutic and Research Act. Instead of implementing best practices to characterize the stem cells responsible for engraftment, two primary assumptions have had to be made. First, the functional stem cells are present in the unit without actually measuring them. And second, minimum testing performed post-processing but pre-freeze, that is pre-cryopreservation, will define the quality and potency of the post-thaw product, allowing it to be used for transplantation. Both assumptions are incorrect, since they have neither reduced the 20 to 24% graft failure rate nor the length of time to engraftment. Whereas public cord blood banks are required to perform minimum criteria testing, there are few, if any, regulations that require private cord blood banks to perform any testing at all. The same assumptions are used by private cord blood banks, but only the basic tests of cell count and viability are usually routinely performed. Other tests are performed randomly. Parents have no way of knowing if the cord blood unit they have paid to be stored will actually perform as intended, if and when the time comes for the unit to be used. Parents should insist on stem cell quality control at periodic intervals to ensure that stem cell quality and potency is stable during the storage period. At the present time, the public is being misled into thinking that if they receive a stem cell transplant, the stem cells will be of high quality and potency, when in fact the stem cells are not even measured. It is obvious that cord blood testing requires a new paradigm, a need for change. There is no scientific, logical, moral or ethical reason for cord blood banks not to measure the stem cells in their manufactured products, and for transplantation centers not to ensure that the stem cells they are transplanting are of high quality and potency prior to use in a patient. Failure to change and implement these better practices is not only a betrayal of public funds, but also of public trust. For more information regarding the published article and how HemoGenix can help improve umbilical cord blood testing, please contact us. Thank you.